So today I will again continue with beta oxidation, the last part of it. And with this, I'll share my screen of my pink board. Are you people able to see this board?
Yes, sir.
Yes, sir. Yes, sir.
Okay. So first thing which we will discuss today. Oh, this table is shaking a lot, so I have to hold it like this. So the first thing which I will discuss today is Carnitine shuttle. So carnitine actually it is a soluble small molecule. Another property of carnitine is that it associates with fatty acids or acyl CoAs actually. So the function is essentially, to transport the fatty acids from cytosol to the mitochondria. This is the function. Fatty acids normally fatty acids take a lot of time to cross into the mitochondrial matrix. So this is not metabolically feasible. Metabolically not feasible because energy requirements are much higher. Are much higher. So to speed up this cannot wait forever till for the fatty acid, it is now come into the mitochondria, and after it has come down into the mitochondria. uh, just hold on. um, if once it has come into the mitochondria, it is going to uh, get now beta oxidation will take place, then energy will come. You cannot wait for that. So this whole process, uh, so this whole process is speeded up by the carnitine shuttle. Essentially, Three components are present. Three enzyme components. The first is the CPT1. The CPT1 means carnityl palmitoyl transferase 1. Carnitine palmitoyl transferase 1. The next one is CPT2. Same thing, carnitine palmitoyl transferase 2. And the third one is a huge name, SLC 25A 20. 25A20. It is also known as acyl carnitine translocase. Translocase. Basically, this is the transport. It's a transporter. So let us look at how it looks like. So let us first divide the screen into three parts. This is the cytosol. This is the matrix. This region is the intermembrane region. In between the membrane, outer and inner membrane. So, cytosol. And what is going to happen? Fatty acyl CoA. Already this is charged. Fatty acyl CoA from cytosol, it will move into the mitochondrial matrix. Now let us draw the enzymes, three enzymes. First enzyme, second enzyme. This is the CPT1. This is the CPT2. And then you have the translocase, acyl carnitine translocase. This is the transporter. Now, here what happens, acyl or the fatty acyl CoA, it binds with carnitine, and with the help of the CPT1 enzyme, it gets converted into acyl carnitine, the compound. Carnitine gets attached to the CoA is exchanged off with carnitine. And now this makes it easy It gets inside, the acyl carnitine gets inside. And now this transporter or this translocase enzyme, what it does is it moves acyl carnitine inside the mitochondrial membrane. Inside the mitochondrial membrane. Now this acyl carnitine is again broken down into, it again becomes acyl CoA. CoA is taken up plus carnitine. The reverse of the this CPT1 reaction takes place. Reverse of CPT1 step. This takes place. And now this carnitine, it's a small water soluble, so a lipid solid moves from here to here, it moves away. Small soluble carnitine. It is lipid soluble, not water soluble. It moves from here and it comes back out. This carnitine comes out. And carnitine again goes here. And this, therefore, it is sometimes also known as the carnitine cycle. It is actually a shuttle to move fatty acyl CoA from the cytosol into the mitochondrial matrix. Is it clear for everybody?
Yes, sir.
Okay, okay. So now let us talk about the next part. Dog dog করছে এটা টেবিলটা। Beta oxidation of odd chain fatty acids. We are still, well, last day we did with even chain saturated fatty acids. Now with odd chain fatty acids. We are talking about saturated. So eventually say 17 carbon, it will break down into, you are supposed to find 7 into 2 carbon acetyl CoA plus 1 into 3 carbon propionyl CoA. CH3CO. So I will show you the structure before I will, instead of writing it. I guess it's somewhere written over here. This is the structure of acetyl coenzyme A. CH3 CO SCOA. This is acetyl coenzyme A structure. Right? This structure was written yesterday. Are you able to see the structure?
Yes, sir.
Yes, sir.
Here there is a CH3 group where my arrow is moving. This is acetyl 2 carbon. One carbon is here. The second carbon is over here. This is acetyl carbon. But if I draw one more line over here, it becomes propionyl. I even did not draw this particular carbon also here. I can simply draw it something like this is also a structure of acetyl CoA. Are you able to understand this? If I draw it like this, I'm not drawing this carbon, any carbon over here. For simplicity, I drew the carbon over here. Is it clear?
Yes,
Yes,
sir.
sir.
Oh, so I have come to this part over here. So acetyl coenzyme A, if I draw it like, just drew a, this is acetyl coenzyme A, and in propionyl CoA, I just simply draw it one more carbon. Are you able to understand? Just one more CH2 is added here. One CH2 is added over here, and one CH3 over here. So propionyl has got 3 carbon and acetyl coenzyme A has got 2 carbon. Is the structure clear?
Yes, sir.
Yes, sir.
Yes.
So just for simplicity, just this, because the more carbon you see written, it becomes very difficult to remember. Therefore, it is better to draw a skeleton like this. These were devised by brilliant chemists and biochemists for simplifying very complicated equation. But this means like this. So you really don't have to bother too much about remembering the structure. Just follow the structure and draw it like this. You will never forget. The more carbon and the hydrogen, where is it? The more you think, imagine, the more difficult it becomes. So now, what this is the so in the end, this is this directly it enters this acetyl coenzyme A. What it does, it enters into the TCA cycle. This enters into the TCA cycle, and you know that one acetyl coenzyme A, 10 ATP. This is the what you get. Now what happens to this bhai sab over here? It can't enter into the Krebs cycle. So I'm drawing it once more. This is the propionyl CoA. Now you require biotin. Biotin is a vitamin. ATP, biotin. They enter. Then a CO2 comes in. It also enters. They are going to add more carbon to this. So the enzyme is propionyl CoA carboxylase. What happens? Carboxylase. And now you're trying to add a CO2. What you do is okay. CO2 minus gets attached over here. Is from one CO2. One hydrogen removes, one carbon is over here. So CO2 minus this particular structure is formed. This is known as methyl malonyl CoA. Methyl malonyl CoA. This name of this compound. এটাকে একটু তুলি ওপরে. Now vitamin B12 will enter. And the enzyme is methyl malonyl CoA mutase. Methyl malonyl CoA mutase. This enzyme will now start working. It needs vitamin B12 as cofactor. And what will happen is this will add up again. এটাকে একটু উল্টে দিলাম. This carbon dioxide gets attached over here. We have flipped this and this comes over here. ওখানে কার্বনটা আসলে কি হলো, one extra carbon has been added over here. Essentially I'm drawing the carbon here. এই পেনের এই জায়গাগুলো দেয় কেন আমি বুঝি না. This is very disturbing. Essentially CO2 minus, which was attached over here, it comes to the end. And therefore, because this carbon, I have drawn the carbon over here. So it is CO2 minus. So you can find that just here you can 1, 2, 3, 4. What you had over here? 1, 2, 3, and the fourth carbon was here. The fourth carbon goes to the end. So this is the structure of succinyl CoA. And you know, succinyl CoA will then enter into the Krebs cycle. It is also, as you know, it is also the intermediate of a Krebs cycle. So half the Krebs cycle will go from the succinyl CoA step. Not acetyl coenzyme is in the first step, but succinyl CoA is just in the circle opposite. So if this is the whole cycle, acetyl CoA is over here and succinyl CoA is over here. So basically the half step will take place. Intermediate of Krebs cycle. So this is clear what happens with the odd chain fatty acids saturated. Yes, no?
Yes,
Clear?
sir. Yes,
Okay.
sir.
Yes, sir.
Okay. Now we will go to unsaturated fatty acids. What happens with beta oxidation of unsaturated fatty acids? But before we go over there, we will discuss a bit about unsaturated fatty acids. How we label unsaturated fatty acids. Unsaturated fatty acid means দানাটাতে ধরে রাখতে হচ্ছে হাতটা। That means they have double bonds. So let us draw fatty acid. এই যে CO, SCOA. Just remember, I'm just showing you the before the photograph of a saturated fatty acid. COSCOA. Fatty acyl. We are starting from the acyl. COSCOA. So this is the fatty acid. So we start drawing. C1, 2, 3, 4, 5, 6, 7, 8, 9. So essentially we are drawing a fatty acid. Now we will introduce a double bond. So this was C1, C2, C3, C4, C5, C6, C7, C8, C9. Now we are drawing a double bond. and this was the ninth, and this was the 10th carbon here. Now I'm again going 10, 11, 12, 10, 11, 12, 14, 15, 16, 17, 18, 10, 11, 12, 13, 14, 15, 16, 17, 18. This is a actually this type of a structure. This is a cis conformation. How do you know it is a cis conformation? Between 9 and 10. This is carbon 9 and carbon 10. This is carbon 9 here, carbon 10 over here. And let us cut this. No double bonds over here. I initially, by purpose, I drew it just to erase it in front of you. Nine and 10. The hydrogens are over here. And you have other carbons attached over here. This is carbon number 8, sorry. This is carbon number 11. As a result of, if the hydrogen is like this and the double bond is like this, because of that steric conformation, the structure of this region, it becomes like this. So, therefore, this is a cis fat. It is not that bad for health. I will tell you why. Now, parallel to this, I am drawing, let's draw it from the top. This is a trans fat. Now, let's draw the same fat. We are having this conformation here. Now, what if it is like this? This is carbon number 9 and carbon number 10. This sort of a conformation is known as trans fat. This is a trans, not fat, trans conformation. It's of course a trans fat. Now, what does this indicate? This means if I draw it, we draw this like carbon 1, 2, 3, 4, 5, 6, 7, 8, Here we come to 9 and 10. The double bond is here. One hydrogen is here. If this configuration comes and the other is here, so this is carbon number 9 and 10. And because it is in this trans configuration, 11, 12, 13, 14, 15, 16, 17, 18. So 1, mm, here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and 18. This what you get is, my friends, called a trans fat. Now, why cis fat is less dangerous than trans fat? Now, I am going to draw this in a very bad manner. Another cis, a trans fat molecule can come inside and stack like this through inside. It will fall in between the grooves. Another molecule can come and it can again stack over here. Result of this is জমে যাবে. They will coagulate and cohese like your fat pieces in mutton, chicken, beef, the fat pieces, it will coagulate like ghee. So you have lots of trans fat, and these things, when they go inside your artery, they will coagulate inside your artery and cause arterial block. The blood will not pass. Those who eat too much of ghee and butter and all these things, and trans fat, churbi, beef lard, pork lard, and those who take too much of these things, very tasty, but problem is that they will go and coagulate in your arteries, and this is not good. But for in the case of cis fat, because of this double bond over here, another molecule will not fit. So this will be in this will stay in a solid form. This will solidify even at low temp at even at higher temperature. Body temperature also it will solidify because of this region. They will coalesce and cohere and they will solidify. And this is the cis fat, because they will not fall into grooves because of this kink over here. Molecule বেঁকে গেছে, it has been twisted, kinked. What will happen? This will stay liquid. So any oil which stays liquid at room temperature is less harmful. That is also same for, we are talking about unsaturated fatty acids. If you look at the structure of a saturated fatty acid, saturated fatty acid like ghee also is high rich in saturated fatty acid. Rated fatty acids are unhealthy. Why? Because there is no double bond. No double bond means no kinks. It doesn't turn. So this is essentially a cis fat. But the moment you have a double bond, and in the double bond, it becomes in a trans configuration, immediately this is very, very harmful. Are you clear about the structure of this cis fat and trans fat so far?
Yes, sir.
Yes, sir.
Yes, sir. Now there comes a nomenclature. We add now after explaining this concept of the double bond, we are now going to a nomenclature. How do we name it? Now this particular fatty acid has is this is in a cis configuration because of this cis configuration. So it is cis. Where is the double bond between 9 and 10? Therefore, it is written delta is for double bond, delta 9, because the double bond starts from the ninth carbon. And how many carbons are there? 18. C 18. And how many double bonds are there? One double bond is there. Is to 1. So how will you call this? Can anybody say? The trans fat? Instead of cis, you write trans. Where in which carbon is the double bond? Ninth
It's
carbon, the
9.
double starts. How many carbons? 18 is to 1. This is the nomenclature. Now we are going, let us go to the beta oxidation of this. Let's start with the trans fat only. Let us go to the three rounds of, for this, three rounds of beta oxidation takes place. So what are who are eliminated? This is gone. 1, 2, 3. Three rounds are gone. And the structure becomes smaller. Beta oxidation takes place. This one goes, two goes, and three go. So one has gone, two has gone, three has gone. So what is remaining? From here, from the 7, it is remaining. So this becomes the new one. Aha. We have reached a blind part of this. So what is remaining here? And therefore this from here it becomes, this is C, becomes the new C1, and the rest of the structure from the 7 it starts. Uh-uh. 1 এখান থেকে পড়ে রয়েছে, 2, the double bond starts right from here. এই গেল, এখানে double bond. So basically this is the 1 carbon, 2 carbon, 3 carbon, 4 carbon. From here 5, 6, 7, 8, 9, 10, 11, 12. So 4 becomes 5, 6, 7, 8, 9, 10, 11, 12. So can anybody tell what is the name of this? This is a cis fat. So what will it be? Somebody say.
This says delta 3,
Cis delta 3, very good.
C12
C12, how many double bonds?
is to 1.
Is to 1. Very good. So you basically you have now understood the nomenclature of how to name fat. This is advanced advanced chemistry. So now there is a, this gets converted into trans fat. Trans fat, and there is an enzyme which is an isomerase. Trans fat over here. So essentially it's the same, 12 থাকবে, but it becomes trans fat. SCOA, 1, 2, 1, 2, 3. So from the 3 the double bond is there, it's over here, it becomes trans. 4, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So this is known as, you can name it also, essentially you can name it like a trans delta 3, same thing, C12, you can name it as trans delta 3 C12 is to 1. But this type of fats are known as trans enoyl CoA. Now we go to the next, the following step. Now if you look at beta oxidation of saturated fatty acids, You see this structure enoyl CoA, the structure enoyl CoA, trans enoyl you are writing, but you are seeing a similar structure here, aren't you? এক দুই দুই থেকে there is a, something DP over here, something like this. You see the same structure over here, isn't it? Yes, no? Similar
Yes, sir.
Yes,
structure.
sir.
So this structure you are seeing now. Now this will undergo the next step of the beta oxidation, which happens. Eh, drawing everything with red. And that is the hydration step. Only this extra step of isomerization takes place. And one H2O gets inside. And then this will proceed. There will be an OH and one OH over here. One OH will come over here and one H will come here. Yeah. Proceeds like beta oxidation of saturated fatty acids. So let us just look at the saturated fatty acids once more. Hydration, what happens? এই দেখো H OH এসে গেল. The double bond disappears. This double bond disappears and HOH comes. Then immediately NADA2 hydrogen is stolen. Oxidation takes place and cleavage starts to take place. And particularly this is repeated once more. So clear?
Yes, sir.
Yes,
Okay.
sir.
Then I'm stopping my
Yes, sir.
class over here. We will again do one more. This next day we will do doing genetics. So we will do genetics next day. I will just check the syllabus. What is the next topic? That is day after tomorrow. I will send you the schedule very fast. Do you people have any questions, by the way?
No, sir.
No questions? Clear তাহলে? পারবে তো করতে পরীক্ষায় in the exam?
Yes, sir.
And one
Yes,
thing
sir.
which I have not covered over here, that is paroxysmal beta oxidation. I've sent the note, just read a few lines about it. I don't think they will give too much about paroxysmal beta oxidation. It is the same essentially, just a few steps are different in that. Maybe one day we will cover a bit in after the genetics, we will cover a bit of this paroxysomes, beta oxidation in the paroxysome. This is all the beta oxidation which is taking place in the mitochondria. This is the main thing. Something also takes place in the peroxisome. So we will also be studying about that. So no questions then. I end my class. Day after tomorrow then, no problem, is it?
Okay,
Okay,
sir.
sir.
Okay, sir.
Okay.